This week we'll be focusing on the eukaryotes. There will be fungi, algae, protozoa, and helminths. It's chapter 12 in the Tortora book. Starting out with the fungi, let's take a look at the term mycology. Myco refers to fungus and ology, the study of, so that will be the study of the fungi. Collectively, they are chemoheterotrophs. It means they're not grain, they don't photosynthesize, they feed on chemicals on others. So dissecting that term, chemo means chemicals, organic chemicals that is, hetero, others, trophs to feed. So these are organisms that feed on others, they don't photosynthesize, and what they do is they decompose organic matter and then they will be feeding on that. In terms of oxygen requirement, they are either aerobic or they are facultative anaerobic. In this table here, you can see the main profile of fungi versus bacteria. The cell type of the fungi, of course, they are eukaryotes. They do have sterols in the cell membrane. The cell wall is chitin. That's something you wouldn't find in bacteria. And the spores, we have sexual and asexual reproduction possible in fungi. And the metabolism, as we mentioned, is heterotrophic, aerobic, or facultative anaerobic. In the fungi, we find vegetative structures. So that would be those structures that are not involved with reproduction. Let's differentiate between molds and fleshy fungi. So the molds, of course, more considered sort of smaller growth. They don't have this mushroom type of fruiting body. But let's take a look at that. For molds and fleshy fungi, they have this thallus, and that's a term that you need to know. That's basically the body of the fungus, the thallus, the body. And it consists of hyphy. That's the plural. Hypha would be the singular. So hyphy, that's kind of filamentous growth that makes up the thallus, the body of the fungus. The mass of the hyphy together is called a mycelium. That's another term you should know. The hyphy, they can either have cross walls or no cross walls. So septate hyphy, they have cross walls. And the cenocytic hyphae, they do not have those cross walls or septa. The vegetative hyphae, they obtain nutrients, while these aerial hyphae, they would be involved with reproduction, so they're not vegetative structures. So here you have the difference between septate hyphae, the ones that have the cross walls. You can see the septum, these cross walls, and cenocytic hyphae, they don't have those cross walls. And here will be a spore, and there you can see hypha, one of them, coming out of the spore. Let's take a look here at aerial versus vegetative hyphae. Let's start over here on this picture. It's not so magnified. You have the vegetative hyphae over here. That would be sort of the thallus, they're making up the body of the fungus. And then the aerial hyphae, those are on top here. And when you, on an Aspergillus niger, if you are magnifying these, what you see, these black dots that are tiny here, they're magnified over here. So you see the aerial hyphae that are involved in reproduction. Let's take a look at yeast. They are fungi, but most people don't really think of them as fungi. And they're different. They're unicellular. They don't have this filamentous kind of growth that you expect usually from a fungus. There are some yeasts that are budding yeasts. They divide unevenly, so a little bud is coming off of our mother cell, and then the bud gets larger and larger until it breaks off, or it may even stay. And then we have a fission yeast. They divide evenly. That's more like what bacteria would do when they divide by fission. There are also dimorphic fungi. They grow yeast-like at a temperature of 37 degrees Celsius and mold-like at room temperature around more 25 degrees Celsius. Here's a nice image of a budding yeast. You can see here's a bud that's really big already, that's still growing. And then here's a smaller bud that's coming off on this side. And here would be a budding scar where the new cell has actually left the parent cell and has left this little bud scar. Here you have an image of dimorphic fungus, yeast-like growth. You can see the yeast-like cells. They look more like the typical rounded cells. And then mold-like growth would be this filamentous growth that you have here with these small hyphy-like structures. Now let's take a look at the fungal life cycle. A fungus reproduces sexually and asexually with the formation of spores. These spores then detach from the parent and germinate into a new mold. Let's take a look at some of the terminology that you need to know. So asexual spores, they are produced via mitosis and cell division. They're formed by the hyphy of one organism. And there are different types. Conidiospores, they are not enclosed in a sac. And you need to know this term. Conidiospores, not enclosed in a sac, open spores. Arthroconidia, those, they're having this fragmentation of septate hyphae. That term you should also know. And then blastoconidia, this term you will probably not hear again. They're buds of the parent cell. Chlamydoconidium, that's a spore with a hyphal segment, also not that important, and this one here is important. Sporangiospore, these are enclosed in a sac. So in the lab too, we'll be mostly considering conidiospores, these here, that are not enclosed in a sac, and the sporangiospores that are in a sporangium, that's a sac-like structure, so enclosed in a sac. So let's take a look at some of these, what they look like. Here will be conidiospores. Here you can see the conidia kind of open right here. Then here will be arthroconidia. In this case, you have this fragmentation. You kind of can see that when you follow the arrows right there. Pseudohyphae, those would be false hyphae. They are kind of lookalikes, but they have these blastoconidia right here. This conidia, spores in this conidium structure. Then here, chlamydoconidium. Again, this one we don't really need that much, but here very important, the sporangiospores. That's a spore case that carries the spores. So again, the two most important ones will be the conidiospores right here, this one right here, and the sporangiospores that are carried in a sporangium in a spore case. Now let's take a look at sexual spores. Now when you talk about sexual reproduction, it typically involves the fusion of gametes that are haploid that then lead to a diploid organism again. So typically when it comes to animals, we have the gametes are egg and sperm, and they fuse together to form a zygote. It's not as sophisticated when it comes to fungi, but still you have sort of these gametes that are fusing together, and it is a form of sexual reproduction. So you have the fusion of two nuclei from two opposite mating strains. In this case, they're just called plus and minus cells. So here, the three phases of sexual reproduction in a fungus will be plasmogamy. That means the haploid donor cell nucleus, so this plus cell, penetrates cytoplasm of the recipient cell, the minus cell. So it's kind of like sexual reproduction in mammals or in animals in general, where you have, fusion of an egg and a sperm, of two gametes. Anyway, karyogamy means that the two nuclei fuse. Karyo refers to nucleus, and that will be the plus and the minus nucleus. They fuse together and form a diploid zygote that is very much resembling sort of animal sexual reproduction. And meiosis is the reduction division. So this diploid nucleus produces haploid nuclei that will be the sexual spores. So these are then the ones that can then fuse together again to form a diploid organism. And as far as nutritional adaptation is concerned for fungi, they tend to grow better in a slightly acidic stage. If you guys would have been able to do the lab on the microbial growth, you would have seen that While bacteria don't like to grow at a pH of 5, fungus, mold, yeast, they really like to grow at a pH of 5, so slightly acidic. And they grow in high sugar and salt concentration, so most likely if you have, let's say, some maple syrup and it has a very high sugar concentration, you will not find bacterial growth in there that spoils it, but you'll find fungus that can grow in high sugar and high salt concentration. They're pretty resistant to osmotic pressure on the outside. They can also grow in low moisture content and can metabolize complex carbohydrates. Some medically important fungi will be Zygomycota, Microsporidia. This one is actually pretty important for medical purposes. Ascomycota, Basidiomycota. So that's these are medically important. Let's take a look at Zygomycota first. That's a conjugation fungus. It has sinusitic hyphy and produces asexually using sporangiospores, and reproduces sexually using zygospores. So it forms when two nuclei of two similar cells fuse. And here will be the life cycle, the exciting life cycle of Rhizopus zygomycete. Here will be in blue the asexual reproduction involving a sporangium, and then the sporangium bursts, releases the spores, and then the spores germinate, form hyphy, and that's the vegetative hyphy. It could stay in this cycle, or it could then go into sexual reproduction, where we are going from vegetative mycelium to forming gametes that go through plasmogamy. The zygospores form, and then we have karyogamy, basically the fusion of these gametes, and that produces a zygote, and then the spores are released from a sporangium again, and that germinates into hyphae, and then we could go either this way or go back this way. Next up on our list of the medically important fungi were the Microsporidia. They don't have a sexual reproduction that you could observe, but it probably does occur in a host. Now, they are obligate intracellular parasites. They do not have mitochondria. That means they are relying on the energy metabolism of their host cells. And so here would be what it might look like when somebody's infected with a Microsporidium. Starting out here with one. The spores are inhaled typically. Usually this is like from the dust, and you inhale something like that. The spore injects the tube into the host cell, and so basically the fungus sort of grows into this host cell, and then the cytoplasm and nucleus enter into the host cell. The cytoplasm grows, and the nuclei reproduce. And then next up, the cytoplasm breaks up around the nuclei to form the spores, and then the spores are released from the host cell, and so forth. And so here, this is what they could look like. This host cell right here with all these little mature spores inside of them right here. Next up, Ascomycota. Those are sac fungi. They have septate hyphae, teleomorphic fungi. That's all what they're usually called. They produce sexually and asexually via the spores. Some are anamorphic. They have lost their ability to sexually reproduce. So they reproduce asexually. They have these conidiospores, and if they produce sexually, then they have ascospores. Their nuclei morphologically similar, or similar fuse in a sac-like fungus. That would be sort of this fusion that resembles, in general, sexual reproduction. And here will be the life cycle of such an Ascomycete. And here, very similar to what we had previously. There you have the hyphae. They're producing conidiophores. That's the stem part here, and then here the conidia are released from the conidiophore right here. So these conidia are released right there. The conidium germinates to produce hyphae. Then you could just keep going in the asexual reproduction cycle, and or you could switch over right here, and the vegetative hyphae produces spores that are then fusing, plus and minus. So if you have plasmogamy, plus and minus cells are formed. and they would have to fuse to form a new fungal growth by sexual reproduction. So we have here these new ascus right